What's up, everyone? This is part one of my conversation with Ernesto Estrada. We will talk about amazing Cuba, how proteins are produced in the cell, and in this context, a drug which is against both bacteria and fungi, intercropping, fractional diffusion, the spread of infectious diseases, semiochemicals, North American tribes, and little plant killers, which are really a problem given the current population growth of Earth. Enjoy. Yeah, Ernesto, thank you very much for having you. It's a pleasure for me. And yeah, I'm very honored to have you here. So let us start where how you grew up, because I find that is very interesting. So you grew up in Cuba, and you told me before that it was one of the poorest districts. And I have sort of like, I want to open with two questions. So one, so you spent a lot of time in UK, and I think that is sort of something where you went ahead in your career and in your life, sort of in that way a better place so compared to where you grew up. But on the other hand, I think in Cuba there's also a lot of nice culture. The people are very nice, it's very warm, and that might be different in UK. So can you maybe elaborate a little bit, so what mattered more for you, or what does matter more for you right now?
Yeah, so first of all, thank you very much, Yang. I'm very excited and happy to be with you here and tell a few nice stories for people. So yeah, I was born in a city which is in the center of the island of Cuba, more or less in the geographic center, which is named Santi Spiritus. It's very curious because it's the only one town with naming Latin in the whole country, because it was one of the first villages founded by Spaniards in 1514. And I was born and grew up in the poorest neighborhood of the town, which is named Jesus Maria. It was very famous by several reasons. But you mentioned culture. It was also very active culture. So my uncle was a painter. There were a few musicians. I have a friend who was a musician around there, and it was very warm. So I knew everybody by name in the neighborhood, and I was playing around with other kids. So we were a product of the 60s. So there was a boom also in Cuba of kids at that time. So we were a big gang, and for good and for bad. And it was very, very nice to grow up there. So the situation from the economic point of view was very, very bad. So
when was it? Was it in the 60s or?
Yeah, I was born in 1966. So
yeah.
It was the Cuban Revolution started in 1959. So there was 7 years later. And but in the 1970s, there was some kind of deep crisis in the whole country, but particularly in poor neighborhoods like mine, this was more more difficult. So I cannot say I was hungry or something like that, but the quality of food and these kind of things, particularly also for when you were a teenager and then you want to have, for instance, a gym, and you cannot have a gym because this was very prohibited for people of my background. And you mentioned what so about my perspective from that, and I remember when I was a kid, I didn't dream with being a scientist or a teacher or academician or something like that. I will tell you one story which is funny. So at that time, there was a guy that goes once per month. It was a guy from the government, and then he was taking the tax for electricity, but there was no automatic system for that. So he had a big bag with the money collected there. So you pay directly in cash, and the guy put the cash into this bag, and then he continued. So I saw this guy as the most important guy in the world because he has a bag full of money. So I was dreaming when I grew up, I want to be a guy like that. So I
Sorry.
I want my kids back. I want my life back. Please tell me, how much is enough? How big does this pile have to be?
But when I was in a nice school in the hometown, in the neighborhood, and particularly at the end of the period of the primary school, I had a couple of good teachers. So one in 5th degree, she was a lady, and then another in the 6th degree in the primary school, and they were teaching science and mathematics. They did some experiments and things like that, and I was really, really captivated by that. So from that moment, I completely was aware that I wanted to be something. I didn't know the name scientist, but I want to do these kind of things. I like to collect plants and study the plants, and to study rocks. So whatever was around, I had this curiosity, which I discovered thanks to these two teachers. Then I moved to another place because in Cuba there was, at that time, there was created some kind of movement, some kind of specialized schools that were particularly tough to enter and tough to be in because they were specialized in science. So the level of biology, chemistry, physics, and mathematics was bigger than in, let's say, in normal schools. But also you have to be intern in that school. So you spend the whole week there. I was 11 years old, so it was very tough for me. So also the level of the education was higher, but also the level of the qualifications was much higher. And then I spent there 3 years until I decided that freedom was more important for me than this kind of specialized education. So then I escaped from the school and I returned to my neighborhood because in that school there was some kind of elite. So even in a socialist communist country.
The school was in Havana, or where
Oh,
was
it was in Santa Clara. It's about 86
okay. Yeah,
kilometers from.
I've been there, I think, even. Yeah. So yeah.
Yeah. It's a famous town because of Che Guevara, and
Yeah.
the name of the school was Che Guevara. But there was a kind of elite among the students there because so from my neighborhood there was only one representative, and this was me. So then the kind of friendship was different, and I liked my pals in the neighborhood, the kind of things that we were doing. So I returned to a normal school, but I appreciate right now that this education, particularly mathematics, was very good, and I was in a team that was prepared for mathematical Olympics games, and that was very, very serious in the socialist countries at that time. So Yeah, it was very good experience, and the levels of friendship and the level of relations, social relations, is different in different environments, even inside Cuba, as you can see. So when I moved first to Spain in 1999.
And I studied in Germany.
And then I moved to the UK. I first spent a year in England, and then 10 years in Glasgow, in Scotland.
Right.
And the social relations are very, very different. So it's a completely different kind.
Welcome to a week in the life of Stop Climb!
And I could say that they are two different qualities. So you know very well social science. So the kind of ties that you have in Cuba in a poor neighborhood is very, very different from the kind of ties that you have in a neighborhood in Glasgow, for instance. I was living in city center in a very, very, let's say, naive neighborhood in Glasgow. But you socialize a little bit with some friends and colleagues, but this is colder, much colder than the social kind of relations in Cuba, where if you need something, you have 100 people that can supply this something to you if they have it. And there is no matter whatsoever if this is the only piece of this item that they have. So if you need an aspirin, somebody has only one aspirin, and he or she will give it to you. It's not thinking, well, this is the only one, because they think, okay, there is a network here, and this network will supply to me. So I used the t-shirts from a friend of mine, and he uses some t-shirt from me, and people interchange some foods, and it was a completely more primitive, naive kind of life.
Sure. Yeah. So yeah, I think in Cuba now there's, I mean, obviously a lot of things changed. Like in particular, if you start like in late 50s or mid 60s or something like that. So now they have, like, for example, universal health insurance. So nobody has to pay, as far as I know. If you can confirm that, like also like in Italy, it's like this. So if you're Italian, you have a health insurance. So there are like interesting things right now which are really advantageous for the people, but yeah, I don't know. So when in the when you grew up, this was not the case, I suppose, right?
Oh, it was. Yes.
Already there. So,
yeah. It's as older as 1959 or something like that.
Ah, okay, fine. Well then, okay. I thought it came a little bit later. Okay, fine.
No, it was one of the implementations of the revolution was this one, and the quality of the health system and in the times I was in Cuba was very good. Now it has been deteriorated dramatically. The quality of the hospitals are in many, particularly in small towns, is very dramatic. And there are many doctors that emigrate, or they are somehow exported by the government to other countries. And the situation now is difficult, particularly in this area. But when I was in Cuba, yeah, you have free health system, and then also you have they call a family doctor, which is different from the GP, the general practitioner in other countries. So it really, you have it's more like a social activist. It's a person, is a doctor specialized in family medicine, but he or she go once per week every home, particularly for elderly people, check the blood pressure, short interview. So these also produce that many elderly people don't go to the hospital because they feel attended, but particularly they don't feel alone. So it's difficult in my neighborhood, but I suppose that in places like in Havana, there are people that can feel alone, particularly elderly people. And these doctors play this kind of role. And it was very important. So the quality of the system in general as an idea was very good. So then in practice, you know that there are many things that doesn't work, particularly because of difficult economical situations at different levels.
Yeah, I get. Yeah, so yeah, you said like when you were very young, like 11 or a little bit younger even, you said, okay, you also wanted to have like this big chunk of money. And you told me before that you even filed like much later a patent, but you did not really get rich because of the patent. Can you tell the audience a little bit about that patent? Because yeah, we didn't get there yet, but yeah, you're a chemist, not really biochemist, but really chemist, and still you went to drug discovery and stuff like that. Yeah, let's maybe start if you find that like with your patent, because I found quite exciting.
Yeah, so I was involved for a few years after I finished my degree in chemistry, and I started to do my PhD in mathematical chemistry. I was involved with, it was first an institute of research at the same university, but the goal of the institute was to develop drugs for human and veterinary health. And the first product that was developed by this institute was a chemical which is an antibacterial and antifungal compound, so both actions at the same time. So I am one of the co-authors of the patent because I was involved in the structural determination, the determination of the structure of this molecule. But there are other people also involved in the chemistry, in the microbiology, in the formulation, etc. So we are a group of people in the patent. So it was very exciting, but it was very difficult because the conditions for any university, even in the first world, to develop a drug is a very difficult task. And by the commitment of these guys, this was pushed ahead until, well, a joint venture was created with a Canadian company. And then the Canadian company and the Cuban government owned the patent for the development of this drug. Mm-hmm. So after a few years, many years, the drug was approved by the health authorities, and is used now both in veterinary and in public health for treating some kind of dermatologic diseases, particularly in immunological compromised patients, but in general for kind of skin bacteria and fungal diseases. And I have the knowledge that it is also used in other countries. But from a scientific point of view, what is interesting is that these chemical act by a completely different kind of mechanism that most of the antibiotics are acting against bacteria and fungi. And apparently, according to more recent experiments, it tackle the DNA or RNA formation or transformation into proteins in the ribosomes of bacteria, one of the two blocks of the ribosome. So there are very few drugs that act in this particular way. And well, I'm very happy to be part of this patent. And yeah, well, it's the Cuban government who owns the patent. They supply the drug with a very ridiculous price for people. I tried to buy the product for having one sample to show, but it was not in the market. So it is a situation, the ridiculous situation that can happen sometimes, that you cannot show even the product that you are author of the patent.
Yeah, so that makes this drug very special because you said it takes sort of some stuff in the ribosome, and that is sort of the reason why. So for me, it was new that there are drugs which both attack bacteria and also fungi, because they're totally different things, right? So is that like very unique, or are there like many drugs which I don't know, which are both antibiotic and also anti-fungi?
No, not many really.
Okay.
And when
So
we were developing these drugs, that every time that you are trying to do something, and this is a message for young people, every time that you are trying to do something, to develop whatever idea, you will find critics. And these critics, some of them are positive and will encourage you with their criticisms, but there are others which are simply trying to destroy your idea. And this is not because they are bad people. It's simply because they believe in another way. So one of the criticisms that the critics put was, okay, so if this kill bacteria and this kill fungi as well, it will kill people. So it kills everybody. It's a biocide, so it will have a great toxicity. So you will never be able to develop this kind of drug. But the situation, and when you see, for instance, the work of the people in microbiology that were working when the product was in the clinical trials, we have to analyze all these pictures. And what happened is sometimes you have the problem in your nail, and then you go to the GP, and the doctor medicate some antifungal compound.
right.
Simply because this is a typical kind of candida albicans that you have there. But the problem is that, there is something I will repeat more than once, and then for every complex problem, there is an answer that is clear, simple, and wrong. So then you have a fungi there, but there are opportunistic bacteria which were living there as well at the same time, and now they see the opportunity to conquer more space for their development. So now you are killing the fungi. So you are killing the competitor of the bacteria. What happened is that they are now more happy. So you are using a drug that puts in a worse condition your medical situation. And it is because there is a combination of these two kind of things. And this was the idea that we were trying to develop, is something that kill both at the same time. And then, now there are a few other drugs acting by the same mechanism. So they have the two kind of activities at the same time. But in the 1990s, when we were developing this drug, there were almost none, to the best of my knowledge. So it was very unique. And the mechanism of action, I was working in the mechanism of action, and my hypothesis was completely different. And it was a more classical kind of hypothesis. At that time, I was using computer and mathematics in order to guess and to simulate what the drug was doing in the biological environment. And again, I was trying something simple and something clear. And as you can see, very recently, I think it was 2005 or something like that, a group of authors found experimental evidence by using X-ray of the drug and the proteins, that they were acting in a completely different mechanism directly to the ribosome of the bacteria.
So there was one paper. So we in the chat before we discussed a couple of papers, and one paper really I found really interesting and also a little bit funny, and I think I also learned a lot of stuff there. Can you refer a little bit to this Aphids paper, how they hop, what is the problem for the crops and stuff like that?
Yes, I have a copy of the paper here, so
I can blend it in, no worries. So that is.
So no, but the story started before this paper, because I developed a mathematical approach that generalized some kind of mathematical operators used in network theory in order to describe practically every dynamic. So this operator is known as the Laplacian, but it doesn't matter right now. What is important is that what is an operator? Well, it's just a mathematical object that, let's say, that controls the way in which something is progressing in a given setup. In this particular case, we have a network, and we almost all know what a network is. We are part of social networks, face-to-face, but also online. We use transportation network when we move by the roads or the trains or by planes. But there are also networks inside us. So there are vascular networks, but there are protein-protein interaction networks, metabolic networks, and many, many networks around. So the internet, the www are examples of networks. So then this kind of operator is controlling dynamics that takes place in the most simple way. So if something is in one particular node, and this node could be representing any entity of this network, this something can go to any of the nearest neighbors of this particular node. Let's put one simple example. So I have a piece of information and I want to share this information with people. And the only way I have, if I want to tweet this, is to tweet it, but then only people which are connected to myself directly in Twitter can read this information. So I can pass this information just through my nearest neighbors. And well, in the case of an airport transportation network, the constraint is bigger, because I can fly only from A to B if there is a flight connection between these two airports or cities. But there are some kind of physical systems in which things fly from one place long away to another place, even if they are not connected. This is particularly true in cases of material science. For instance, when you have a chemical which is absorbed over a metallic surface, it is moving just from one place to the nearest neighbors, but from time to time, it flies to 3 neighbors apart, 6 neighbors apart, 9 neighbors apart, or so forth. So the typical operator I was mentioning that has been since Kirchhoff, the 19th century, it has been around, only taking into account these, let's say, transportation to nearest neighbor. So I make a generalization to consider these flights to farther away nodes.
So I mean, but that would be also described, let's say, by fractional diffusion equation and stuff like that, but you haven't invented this, or did you? So what I know is like this fractional calculus and Levy flights and Levy jumps, and then maybe you have like a combination of, yeah, maybe then I have to explain what I'm now talking about, like whatever combination maybe of subordination and long-term jumps, and then you have effective exponent how things spread. So so now you started with your Laplacian, and you have this generalization. Can you link this to what I just said, and maybe also explain better than I did, because I did not?
Yeah, okay, so we are diverging a little bit from the Aphids, but I will take a
Why not?
Yeah. So yes, the fractional calculus is something that sounds strange even for scientists. But everybody here in the school studied the derivative. So then you find the derivative, and then you know that this is the rate of change of something with respect to time. And so we analyze, for instance, the speed of a car by taking the first derivative of space with respect to time. But then we make a generalization of that, and we talk about acceleration. And the acceleration is the second derivative of space with respect to time. So we take d2s dt2, and then we obtain this acceleration there. And then we can make a generalization to the third derivative, fourth derivative, fifth derivative, etc. But if you listen to what I am saying just that first, second, third, fourth derivative. All of these numbers are integers. And mathematicians are curious people. They ask the question, what happens if I take the 1.5 derivative of something?
Mhm.
What happens if I take the 0.5 derivative of something? And there are a few definitions of these derivatives. But basically what you do for our listeners is the following. When you take the second derivative, you simply take the second derivative of something. But now if you are taking the 0.5 derivative of something, you take the derivative a little bit more. Let's say you take the first derivative, and then you recover what you lose by integrating. So you have an integral differential equation or formulation for this derivative. One of these derivations is known as the Caputo derivative, which is more physically appealing. So it has some of the properties that physicists would like to see in this fractional derivative. And as you will say, if you take, for instance, a diffusion equation, which is fractional in space, you obtain this kind of anomalous diffusion that take into account these long-range interactions between the particles.
Oh boy, fractional calculus in a nutshell. So it has something to do with diffusion. Let's assume you put ink in your bathtub, and then the ink will sort of diffuse around, and the diameter, the expected diameter, scales with the time to the power of 1/2. So meaning with the square root of time. So that is called normal diffusion. Normal diffusion. Okay. So these aphids, they cannot stay at some point every time, because at some point there's no food anymore. So what they do is they go around, find food, and then they fly somewhere else. And if there's no food, then they fly somewhere else, and so on. And the displacement can be written as t to the power of alpha divided by beta. And alpha and beta can have multiple values. And that is usually referred to as, could be, for example, super diffusion. And fractional calculus sort of finds a way to address this alpha divided by beta in equations, how this works. So fractional diffusion, so what is the alpha? So the alpha, let's assume this aphid is just like staying at some point. So the time where it stays comes also from a distribution. And let's say the probability that it stays a certain time here scales like t to the power 1 plus alpha. So that is the alpha part. So that has something to do with a waiting time. So of course it's a model, but this is a waiting time, how long this aphid spends at a given place. And this jumps, let's say it jumps from A to B, so that can have different magnitudes. And the probability then is modeled as the displacement to the power of -1 plus beta. So meaning alpha is for time, and this beta is for the displacement. And if you go back, you see here, they are here together, right? So, and you can show that you have to divide them in order to get this displacement. So now, I haven't explained actually fractional diffusion in equations. So let's have some equations. So if you have normal diffusion, you have, as I said, something like the displacement scales as t to the mu, and mu is 1/2. Okay, that is normal diffusion. If you want to have the probability p that at some place x at some time t, there's some ink, then there is a partial differential equation for this to describe exactly this probability distribution function, where this ink is sort of distributed at a given time. And for the normal diffusion case, so this equation goes like this, that you say, you just like have the derivative with respect to time. And here you have dx squared plus dy squared plus dz squared of this p, of the distribution. And there's also a name for this. This is the Laplacian operator. And yeah, that is normal diffusion. So what is now sort of super diffusion or fractional diffusion? So that is very similar. So fractional diffusion, this is a generalization. Now we come back to this alpha beta. So the governance equation is just like dt to the power of alpha equals sort of this Laplacian to the beta half of p. So that is the fractional diffusion equation. And as I said, alpha belongs to the waiting times, how they are distributed, and they have to be distributed in a power law fashion. And beta belongs to how far the jumps are. And the exact distributions I haven't given here, or did I? Yeah, that is here. The distribution of this guy here is that for the displacement, and the distribution of the waiting times are this. And if you plug this all in, then you can show that this is actually how current description is. And as an example, so Ernesto obviously also gives an example. He uses a slightly different method, but the main idea is the same of this partial sort of, it's the partial differential equation, and you have like powers of differential operators. In this case, it's dt to the power of alpha, and here also these, this Laplacian to the power of beta half. An example would be for human travel. Human travel, then roughly is described by alpha is 0.6 and beta is also roughly 0.6. And that would lead then to a displacement of t to the alpha divided by beta, and that would be t to the 1. That means that the displacement for human travel scales like the displacement for normal diffusion square. So it's much faster, and that means like if an infectious disease is spreading all over the globe, it will reach like almost all places.
So this is one way to do things, but I could say it's not the natural way of doing things. So if you have something like the Laplacian operator in the graph, so I am taking in a network, and so how you make a generalization of these that take into account these flights from one place to the other. Let me clarify something for a specialist. So in the continuous, if you have a continuous space, you really have these Levy flights, because in the continuous space, what you have is that the probability to having these flights for a certain distance is identical for every single distance of the same size at every place of the space. So the space is homogeneous. But in a network, it doesn't happen. There are regions in which this probability of jumping between three nodes is much bigger than in other regions to jumping between three edges as well. So then you really don't have exactly the same definition as a Levy fly. But we can say that this is the analogous of the Levy fly for the continuous. So for the discrete, sorry. Yeah. So then I make this generalization. And when you do a generalization, this is another message for young people, is you have to recover what you are generalizing as a particular case. Well, in the case of the fractional Laplacians, and you can take a fractional Laplacians in the discrete as well. So there are a couple of, well, there is one Mexican author and another Colombian one. They have taken the fractional powers of the discrete Laplacian. And what happened is that in this particular case, when you take the fraction one, you recover the fractional Laplacian as usual. So this is a good generalization. So our generalization goes in another direction. It takes the possibility that this particle is not only hopping to the nearest neighbor, but to the second nearest neighbor, the third nearest neighbor. And then you have a set of Laplacians that you can transform in a mathematical way to give a probability that this hoax occurs with certain kind of frequency law. You can say, okay, this decays exponentially, or this probability decay as a power law, etc. And there is an advantage of this respect to the fractional powers of the Laplacian. The fractional powers, which are the roots, okay, if you take one metric raised to 1/2, what you are taking is the square root of this metric, okay? Okay, if you take the square root of any number, you always obtain one single number, okay? Well, in case that you have square root of a negative, it could be complex, or it could be a real number. But if you do this for a metric, you have a problem. Not always the root exists, and in some cases, the root is not unique. So there is a condition that you need for the roots to exist and the roots to be unique. The Laplacian fulfill this condition, okay? So the Laplacian is a semi-defined, so positive semi-defined metric. So all the eigenvalues of the metric are non-negative, and then this square root or any root exists. But if you take the adjacency operator, which is the second most important, or I could say arguably the most important operator in network theory, these roots doesn't exist. So you cannot apply this formalism, for instance, to epidemiological models, because the epidemiological models are controlled by the adjacency operator and not by the Laplacian operators.
I see. So,
however,
yeah, let me, if you allow, let me interrupt you again. So,
yes.
So when you were talking about that sort of this long-term jumps by the Aphids, we are not still there, but maybe we are coming to that point. And I also thought, okay, why you don't put this in the topology and have a simple dynamics? Let's say you have like long-term connections, like a small world, and on top of that, and if this is representing the dynamics very well, if you have a simple dynamics where you have like longer range connections. So the question is, so what I didn't get yet is, why you develop such a complicated thing on a trivial topology and not just like change the topology to something non-trivial and apply some simple dynamics? Or is that?
No, there is an equivalence, but you have a but here, and the equivalent is that the transformed Laplacian operator, which is not complicated, it's very natural and it's very simple, are zeros and one matrices like, well, reduce it to zeros and one matrices like the adjacency metric. But you can then represent this as a new graph, as a new network in which you have some kind of connections. But these connections are weighted. And this weight depends on the original topology that you have. And this is completely different what you were saying, that you do some kind of long range connection. And this is a big mistake in the literature, particularly people that are studying brain networks. They say, okay, so there are long range connections here. No, these are short range, because once they are connected, they are in a short range connection. Even if they are separated very long away in space. One thing is in space, you and I are now connected, but you are in Germany or in Switzerland, and I am in Spain. But we are nearest neighbors. We are not now transmitting a long range information, but a short range one. So it doesn't matter where the space is in the topology. What matters is whether you have a connection or not. And here what happened is that if you made this reconnection as you were suggesting, then of course you end up with a complete graph. Everybody can move with the same probability to any place, and this is meaningless, because if we go to the case of the aphids in the crop, if one aphid landed into one particular plant, the probability that this aphid explore the closest neighborhood around this plant is much bigger than the probability that it goes and fly 10 meters away or 100 meters away from this particular plant. So the biologist has found that the aphid first explored the crop field and determined by the color and also the smell of the plants, which are the good plants for eating. So it lands in one of these plants, but then it explored. It didn't go directly and eating this plant. It explored. It is tasteful, it's nice, and then it goes around and see other plants because now I am in this neighborhood. It is not nice, and then it fly. And this fly decay with a power law probability with the distance, with the geographic distance, with the separation between these plants.
Yeah. So, but that is sort of exactly what people study in optimal foraging. So an organism looks for food, and then typically if it cannot get enough food, or if the environmental local condition is not good enough, then if it can, sometimes it cannot jump or cannot fly, then it will sort of migrate somewhere else, right? So that more or less I'd say all animals, or like maybe all species, or us included, are doing. So in that way, the aphid is not very special. So
no, but there is something different here when you are mentioning and what I am saying, because suppose the albatross, okay?
Yes.
So the albatross is just flying around a piece of the sea, and then if there are no fish there, so it can move away in another direction. So this is the kind of exploratory movement, and this is described by Levy flights. Why? Because it is moving in continuous space. It is moving in the air. So if you take elephants in the landscape, they are moving in continuous space. So it can be described by a random walk with Levy flights. But now you have plants, and the plants are fixed in given positions. So there is no continuous space in which the aphid is moving, but the aphid is moving in a discrete space. And this discrete space is formed by the position of the plants in the crop, or in the forest, if you like. So then now the fly cannot move, let's say 10 centimeters away from the plant, because it will be in the ground. It can move exactly the distance to which another plant is, a nearest neighbor, for instance, or a second nearest neighbor. And this is what the new Laplacian operator controls. How one particular, this is, for instance, what happened in atomic systems. The atomic system is not a continuous as well, because you have atoms. And then if you have an electron cloud in this atom, it cannot move to the middle of two atoms. This is impossible physically. It can move from one atom to the other. And this is a discrete space. So in the discrete space, we don't really have these Levy flights, according to the mathematical definition, all right? So we have this kind of sort of Levy flights, but we have to have into account that the probability of moving between two particular nodes separated at the same distance is not the same as moving between two other nodes separated at exactly the same distance d, okay? So this happens in the continuous space, but it doesn't happen in the discrete space. So let me go to the aphid problem. Because this is very important piece of work. So so for the year 2100, so in 80 years, the population of the world will be between 9.6 and 12.3 billion people. So it means that we have exactly 2 to 4 billion people more than what we have today. So this represents that we have to duplicate the food production. So in reality, we have to multiply 100% or 110% the food production in relation to what we were producing in 2005. Then there is one possibility, which is taking more land. Okay, but we know that we are now constrained by environmental problems, the global warming. And then what happened is that if we continue destroying the forest and the wildlife, we will have bigger problems. Again, we don't have a simple, clear solution for a complex problem. But we have something that put worse the situation, and is the pest. So I have here some data, but the pest pathogens produce losses, for instance, only for rice, maize, and wheat, that has increased in the range of 10 to 25% of losses in the last few years because of the global warming. Indeed, the pest, particularly the aphid, has displaced 2.7 kilometers per year to the north. So in the past, the problem of these pests, particularly aphids, were more the tropical countries around the tropics. But
so just to understand, so the aphids, they hop around, they distribute sort of, and they bring the pest, which is then bacteria, I think.
They're mainly viruses.
Okay, they bring viruses to the plants, and then the crops are dying.
You're probably familiar with aphids, sometimes called plant lice. They're hordes of tiny soft-bodied insects swarming your rose bushes and killing your green beans. But they also do some stranger things to help themselves survive. Things like pooping sugar and cloning themselves and giving birth to pregnant babies. The 4,000 plus members of the Aphididae family infest plants all over the world, and lots of people think of them as pests because aphids suck. Literally. They use their long, slender mouth parts, which work kind of like a straw surrounded by needles, to pierce tender plant stems and suck sap from the phloem, the tissue that ferries sugar and other nutrients through the plant. That sap is a plant's energy source, so a large colony of aphids can damage or even kill a plant by sucking it dry. Not only that, but when an aphid taps into a stem, it injects a tiny bit of saliva and can transmit potentially deadly viruses between plants. They're basically like the mosquitoes of the plant world.
And one other question to understand the significance of the entire thing. So these aphids and the pest which they bring, are they like dangerous for any kind of crops, or is it special sorts of crops?
Yeah, that's a very good question. So there are aphids specialized in different kinds of crops, but okay. There are many different kinds of aphids.
It is very tough to find cute different Aphididae, but I found cute Cicadillidae instead. And anyway, both are hemipteras.
Meet Marvin. Marvin is a very cute leafhopper nymph in the family Cicadillidae. Marvin's a bit of a show-off. Put your tongue back in your mouth, Marvin. Mildred is also a leafhopper baby, and she's very proud of her new dress. Good posing, Mildred. We like your dress too. Ernesto got the fancy class photo package. But Ernesto gets a little nervous on class photo day. Don't worry, you look fantastic. Now, Demia is a plant hopper nymph, a close relative of the leafhopper, but in the infraorder Fulguromorpha. You may have noticed something about her choice in accessorizing. Thank you, Demia. Yes, that's what I was talking about.
And you can almost find groups of aphids attacking most of the food. But for the most important ones, which are maize, wheat, and rice, we have aphids which are moving a little bit more to the north at the rate of almost 3 kilometers per year. going to the north and the south, going to the poles. And this means that they are conquering more and more land. So what is the simple solution? Using more pesticides. Okay, so we know what happened with the pesticides in not only for the environment, but also for human health. So there is a very old solution for this problem.
Biochemistry, genetic modification. What about that? Not very old.
Well, this is another possibility, and
I'm not saying that this is also what we should do or that is healthy necessarily or something, but obviously that if you speak to a farmer or so, they would probably suggest that.
I think the solution, there could be not a unique solution. There could be a combination of solutions. Mm-hmm. This could be part of the solution. There could be necessarily to use more, in certain situations, some kind of pesticides. We hope that there could be biodegradable pesticides or more human healthy pesticides in the future. And there is a third solution, which is very simple, is very clever, and is very old. And this is so old that it was used by the Iroquois Indians that were living in the region nearby today's New York, New Jersey. And they talk about the three sisters. And what were these three sisters? Well, it was a combination of three crops that they put always together. It was maize, some kind of faba beans, and a third one, I don't remember right now, but they put the three together because they realized by empirical evidence that when they put the three together, the three survive healthy without pests.
Nowadays, when we plant corn, we usually plant it by itself when we plant it in rows, but Native Americans actually would not do that. The traditional Native American method used by Iroquois tribes is a little bit different, and that's what we're going to show you today.
So the Native Americans would not plant their corn by itself, and they wouldn't plant it in straight rows either. Instead, they would plant it in clusters together with two other plants, two specific plants actually, beans and squash. And the really cool thing is that those three plants actually work together to supply nutrients to each other, to protect each other, and then the foods that they produce, squash and beans and corn, actually work together to provide more complete human nutrition, which is really pretty amazing considering that the Iroquois didn't know anything about amino acids or complex carbohydrates or things like that, and yet they found and implemented this incredibly ingenious solution that supplies complete plant-based proteins and really great nutrition. So that's pretty darn cool. So here's the way that works. Corn is a grass. Corn needs huge amounts of nitrogen to grow. The way we solve this problem in the modern world is that when the corn gets a little bit higher, we'll put nitrogen fertilizer down the middle of each row, and then the corn roots will reach out, find the nitrogen, and then have the energy that they need to grow. Of course, the Native Americans did not have nitrogen fertilizer, so what they used is natural fertilizer through the bean plant. Beans actually pull nitrogen out of the air and put it into the soil, and so that supplies a natural nitrogen fertilizer for the corn. Pretty darn cool. Meanwhile, the corn also helps the beans, because the corn has a corn stalk, and the beans need something to grow up and twine around to provide support. It's a vine, and so the corn stalk provides that. So the beans help the corn helps the beans. Meanwhile, what the squash does is the squash plays defense. Squash has very large and broad leaves, and it grows out all over the place. Those large, broad leaves help to retain soil moisture, and then they also prevent a lot of weed growth because the sunlight can't get to the weeds, so that's pretty great too. It gets even better than that, though. Raccoons love corn, and they will totally raid your cornfield, and they would totally raid an Iroquois cornfield, but they can't, and I'll tell you why. Squash barbed wire. Squash vines are actually barbed. They have little prickles on them, and the raccoons hate walking over that. They hate it. They won't do it. If you have a corn patch that's surrounded by squash, the raccoons won't cross the squash and go eat your corn.
We know today what happened, and it's chemistry, is that some of the crops produce semiochemicals that repel the aphids that tackle one of the other crops. And they do this in a symbiosis. So now what happened is that if you put, for instance, faba beans and maize together, then the semiochemicals of one repel the aphids of the other and vice versa. So you have two crops in the same place without using absolutely any unnatural pesticide, and both will grow up in a healthy way. So there are empirical studies. There is an institute in Kenya and this kind of technique in agriculture is known as intercropping. So you intercrop more than one crop in the same place, in the same field. So they have determined it empirically that you can put, for instance, maize and small plant, which is a sort of herb, in the middle. So you can spread in the middle, and these produce semiochemicals so powerful that then all the aphids go away from the crop field. Mm-hmm. You can even put a little bit more things, like you can put some kind of other plants around that produce semiochemicals that attract these aphids. These are the traps. So now they go out from the field and go directly to these other plants. So you have the aphids localized, and this is a very, very clever idea. So why I was interested in this? Yes, please.
Yeah. So yeah, just like I think that sounds like yeah, very smart. But on the other hand, it also sounds like this is why we didn't do that in the first place, because in the first place there was no monoculture, and we didn't have all these problems that one single species sort of yeah, destroys one kind of crops. So one could also see it like this. Now we go back from the monoculture to like whatever biculture or something like in the middle to compensate for the problems we actually have imposed to environment or to nature. But of course, when I'm saying this, I totally ignore that we have this pressure to produce more and more, as you just said, for the increasing population. So in a way, I mean, I also don't have like data for that, but probably it is for us just like also impossible that we would go back away from monoculture or from mass production, from non-optimized food production when it comes to plants. So and that way, what you just like saying is probably one of the smartest solution, which avoids then all kinds of drawbacks if you include sort of whatever CRISPR-Cas, the biochemistry, genetic modifications, and pesticides on top of that.
Yeah, well, there are a few things. There are of course many things in your comment, but so you don't need to go to let's say to produce two different crops in the same place. What I'm saying, for instance, for maize is a small herb that you can put in the middle, and even this is, I could say, has no use apart from being expeller of these semiochemicals. So you will be growing maize, corn alone in this particular field. So there is no two crops there. So there are many different combinations, and there are many works in the literature. There is a vast literature. And when I started in this field, I thought like you, like a first world country scientist. Okay, so this is a solution for third world countries with small productions at the local level. Well, I was wrong. And of course, when the production went out for this mass production, people invented the tractor, invented machinery for all of these. So the Aborigines don't have these kind of machines, of course. We invented all of them. Well, this has a lot of advantages and a lot of disadvantages as well. But we invented the necessary tools for making the mass production. So now we can invent a new kind of tools to make this intercropping production at industrial level. And this is something that people had already, engineer has already stated in some way. And so why we don't use these? Well, I have one anecdote. I don't have the story, but the history, but I have this story. So my mother-in-law is a farmer, and she has been growing for her consume a lot of different things for more than 60 years. And her sons and daughters were criticizing very strongly her because she was not very organized. She put corn together with potatoes, sometimes some flowers, and all of these together. Mm-hmm. And well, this is not very aesthetically appealing. So, but then she said something about pests. Well, I know that pests are keep it away if I do it in this way. Of course, she didn't know how to explain that, but she has 60 years of experience about this. Mm-hmm. And I was just listening to the conversation, and I learned the lesson. Indeed, if you look at the acknowledgement of the paper, is dedicated to my mom-in-law because of 60 years of intercropping. But then when we go to this social idea of what we think about modernity, modernity was thought as, okay, so conquering more space and living more productive and all of these kind of things that we already today know that they were damaging the environment, they were damaging our health, etc., etc. So a vaccine against this is a little bit of less arrogancy and a little bit more modesty and learning from the past. So, and this is one of the lessons. I'm not saying this is the global solution, but it could be part of the solution. And why mathematics has something to do with this problem of intercropping and aphids. So what I observed there was the following. In the practical areas where intercropping was used, they use always the same scheme. One line of the crop, one line of the, let's say, trap, another line of the crop, another line of the trap, and so forth. The wall was always one line of each. In some cases it was in a square field, some cases it was a more rectangular field, but always the pattern would repeat. So the question for a mathematician here is this optimal? Is this the best? Is this based on the experience? Of course, there is a reason. It's easier to how to say, to grow the crop and then or to collect the crops at the end, etc., etc. But for a mathematician, you have to ask the question: is this the optimal pattern? So we need to put a model in such a way that we were able to reproduce the experiments, and we were able to reproduce experiments for mice and this trap together, in which we have the crop field with the plants, with the crops, and then we have the traps located in different arrangements. And then we have the aphid with landing in one particular plant and the probability of moving around. And then we analyze how this virus propagated by the aphid was spreading across the field and damaging all the field. So we did all of this simulation, we reproduced these experiments, and now we were having, let's say, a mathematical formalism to make experiments in the computer. And these experiments were, okay, let me change this kind of arrangement. Not only in these lines, okay, so let's put the lines along the rectangle, across the rectangles. Let me spread the second crop in a completely random way. Let me put in like a chessboard place, etc., etc. So what we found is that, okay, lines are not optimal. We're not the best solution. So there are other solutions, including the random distribution of the second trap, that improve dramatically by the 300% the delay in the propagation of the disease across the crop. Also localize the damage into one region, such as that you can just remove this part immediately and the propagation doesn't continue across the crop.
Did you
yes.
find these constellations? It reminds me on lattice optimization. I can give you an example also for that. But did you find this in a systematic way, or was it like that you had like the mathematics described, let's say, the equation which gives the result of how much damage takes place, and then you sort of brute force and sampled some parameters, or did the mathematics also give you guidance to actually, so could you just like whatever set a derivative to 0 and then you solve for the optimal solution and then you had multiple solutions, or like what kind of approach is that?
Yeah, so here we have constraints, of course. So, okay, so the constraints are you cannot change the way in which the crops are cultivated very much. And then you don't have a continuous of solutions. So making an optimization here, as you well mentioned, could be impossible because you will find many solutions which are completely impractical. So what we select is a few kind of clever arrangements. I don't remember exactly the number, but maybe 5 or 6 different arrangements that doesn't change very much the way which crops are cultivated today. And then we test for a few epidemiological parameters of these different arrangements to see which of them is the best. So I'm not talking about the optimal. It could be a possible better solution to the one that we found, but we found that at least by analyzing these very few examples, the one that is most used today is not the best. You can improve very much. And for instance, for the random spread of the second crop, in the case of, it's not necessarily a crop, as I say before, and then if this is a small herb like this one, it's easier to spread this randomly even before you plant the crop. So you spread them randomly and then you put the crop. You don't need to collect this because this is just like a herb growing there. And it's easier and it's much better than the one in which you put into the line. So this was our proposal, and then I'm now in contact with some researchers in the experimental side in order to test some of these in experiment and in certain kind of different plants. Because for instance, you can use this, for instance, for coffee. And then the intercrop with the kind of coffee that has been used, for instance, in Brazil, is a palm tree. It's a kind of palm tree that exists in the tropical areas, like in Brazil, it's also in Cuba. And of course, why you have to put this in a particular line? You can spread across the field in some different other ways, and you can evaluate in this field whether this improve or not the productivity, which is damaged by the aphid propagation.
And you said, so that has been or is implemented in Kenya, or?
No, not our particular approach. So they have an experimental field there, and in which they try. They basically do a very important work, which is using the nice combinations of plants. So if I tell you, for instance, this kind of fava beans, what I can to use in combination with it in order to avoid the pest. So they do these kind of experiments, and they always use the same kind of line arrangement. And what we are proposing is a change in line arrangement. And then I discovered many things that in a previous work, in a collaboration with other co-authors, we studied the propagation of the aphid into the crop fields. And then we were interested in a very simple question. What happens if I take a square crop field or a rectangular crop field with the same area? And what we saw is, well, nobody has studied that, except that in the 1970s, in experimental crop field statistical research, people were aware that when they were trying to reproduce the propagation of a disease, if they change the shape of the crop field, the results were not consistent. So what happened is that, okay, so they always in the experiment put a square crop field, they put the plants there and they analyze some statistical parameters. By some reason, sometimes they could not take a perfect square and they put a little bit of rectangle with the same area, and the results were not reproducible. So they simply fixed, okay, we need to use a square crop fields. But what was the reason for that? And then when we investigate this, and then we discovered that elongating the rectangle field, which from the network theoretic perspective is very interesting because you have some kind of networks over there. What happened is, for instance, that the epidemic threshold, which is a parameter that tell you whether this infection will become an epidemic or not, is displaced dramatically to the right. So increase the epidemic threshold simply by elongating the crop field. And I commented this to my mother-in-law, and she said, yes, of course, I know this since I was a kid. So
that's,
now you have a mathematical reasoning and a way, if you want to try how much elongated, then of course it's not a simple solution because you have, this is an industrial kind of setup, but you can play with it. And I think that this is the task of a mathematician, to provide tools that solve these practical problems. Even if I make an attempt to obtain a solution, it's not the solution what is important. It's the tool that we are providing, because there are other experts in these particular areas which are the guys with the expertise to solve the problem, but using our tools. So this is our goal.
This was the first part. So in the second part, we will talk about social imbalance and how to predict with that wars and how to measure conflicts. And there's also a third part where it's about long-term damage, long-term organ damage in COVID. Enjoy.